Hey, biology. Welcome to our new unit. This is a unit on DNA, okay? Deoxyribonucleic acid. Now, we danced around DNA a while now. We've talked about it with chromosomes. We mentioned it when we were talking about cells last semester. But this unit is directly talking about DNA in depth, okay? We're going to learn a lot about it. Probably by this point in your life, being freshmen in high school, you've heard of DNA. Right? Not just in my class, you've heard about how it carries genetic code and all that good stuff. We're going to look today at the structure of DNA, like what it looks like. Pay no attention that it says 2 right here. I took out part 1. So anyway, this is where your student notes start. This is what you should be using, these videos you should be using to fill in your student notes. And not just fill in the blanks. You need to watch and listen. I promise you I'm going to tell you more than just written down. So we talk mainly about DNA as being what composes chromosomes, right? When we make those little chromosomes like we learned about in the last few units, it's made of DNA, long strands of this DNA. So when we look at DNA itself, it's composed, it's a long polymer. Remember, poly means many. We've talked about this a couple times. It's made of these small building blocks, monomers, mono means one, called nucleotides, okay? These nucleotides. And you may remember way back a while that we talked about nucleotides in like unit 1. But if not, it's okay. We're going to see more about it. So these nucleotides chain together, they link together to make DNA. There's 4 types of nucleotides that we're going to see. Now, when we look at a nucleotide itself, and this is kind of getting into some chemistry that you'll learn more about next year, but that's creeping up on us. Each nucleotide has 3 main component parts, okay? And don't dwell on these too much. We're going to look at them and move on a little bit. All right, there's a phosphate group. There's a phosphate is made mainly of phosphorus and also oxygen. There is a ring-shaped molecule called deoxyribose, okay? And that's where we get the name of the D in deoxyribonucleic acid, DNA, deoxyribonucleic acid, okay? So this deoxyribose is then connected to this nitrogen-containing base. Okay, it's got lots of nitrogen. Here's the chemical makeup for it if that's your cup of tea. Scientists first believed that DNA was made of equal parts of these 4 types of nucleotides that we're about to see. However, as more and more research is going into it, and we're going to look at some of that history of the research, we've seen it's not always an equal part. Okay, and there's a way that they group together. In 1950, so really not that long ago in the grand scheme of the world, about 70 years ago, a guy by the name of Erwin Chargaff changed things by analyzing DNA, lots of different organisms. Okay, he looked at things like Streptococcus, which is a strand of bacteria. He looked at yeast. He looked at a herring, which is a fish, and at humans. And he started seeing the same 4 bases, which we're going to talk about in a minute, of DNA. Okay, these nitrogen-containing bases. Now, the phosphate groups and the deoxyribose all were the same, but these nitrogen bases were different. And he found 4 different types, but they were consistently in all living organisms, okay? And the proportions of the 4 bases were different. So it wasn't like in Streptococcus, there was 25% of one kind, 25%, 25%, 25%. Instead, there was a large variety, a difference in them, especially like humans. You can kind of see some difference here. The proportions were different, which made him realize, okay, it's not just equal makeup of these things. There's something else going on. What he found was there was an equal amount of one of the nucleotides called adenine, and it equaled up with another one of the nucleotides called thymine, okay? And then there was 2 other kinds, cytosine and guanine, right? And they were equal parts. So he came up with something called Chargaff's rules. Chargaff's rules said that in all living things, in this DNA, adenine, the amount of adenine was equal to the amount of thymine. And the amount of cytosine was equal to the amount of guanine. Okay, this led to help us understand how DNA structures together, and then we'll see later on in this unit how DNA functions, how it sends out the genetic code for all living things. Okay, I'm going to give you a really good thing, and this is why you need to watch the videos. How do we remember this, that adenine and thymine go together? Apple in the tree, car in the garage, okay? Adenine and thymine go together, apple in the tree. Cytosine and guanine go together, car in the garage. Apple in the tree, car in the garage, okay? A and T always pair up, as we'll see much more of this in this unit. And cytosine and guanine, car in the garage, they always equal up, okay? So if we're looking for the structure of DNA, we see what it's made up of. These nucleotides that are pairing together according to Chargaff's rules, adenine and thymine, and then also cytosine and guanine. Two guys by the name of Watson and Crick, which we'll look at a little more in depth in some videos and things. Watson and Crick, they're very popular in the world of biology. They developed the first accurate model of what DNA's 3D structure looks like. Okay, and you may have noticed in my room before, there's some blocks, these connect blocks hanging up above, kind of over by my desk. This is the DNA structure that they first came up with. The thing about Watson and Crick, there's some controversy that goes on now, is that they used previous work from other scientists, sometimes not really given credit. We'll talk more about that later. But they used some of this other work, and they hypothesized that DNA was structured like a helix. And a helix is like if you had a ladder and you twisted it around on itself. So you might oftentimes hear that this double helix structure of DNA looks like a twisted ladder. Okay, it kind of causes it to look exactly like the thing that's in my room. So next time you come in, pay attention. So Watson and Crick came up with this. They used other scientists' work, sometimes without giving credit where credit was due. And one of those scientists was named Rosalind Franklin. Rosalind Franklin is a sad yet remarkable story. In a time where women really weren't being given credit in the world of science, especially things like biology and genetics, Rosalind Franklin did some amazing finds. The sad part is she didn't get credit for it until much later on. She had I call it partner, but she kind of did all the work, and Wilkins was the guy that got a lot of the credit because he was a man. Let's be real about what was happening at the time. That's not fair, and thankfully the world is beginning to change, if not already, kind of farther on. We could debate that at different times. The Rosalind Franklin video you'll see posted below here is something that hopefully open your eyes to some of the contributions that never got credit along the way, people like Rosalind Franklin. Anyway, these two people used X-rays, okay? X-ray crystallography is what it's called, but they used X-rays to actually take pictures, and you can see one below right here of DNA. And you can see this structure. Imagine if you're looking down at a staircase or a ladder that's been twisted, you will see these structures going down. It might even look like an X from up above. So they suggested that DNA had this helix shape. Also, the work of these other scientists led to Watson and Crick coming up with that DNA model. This is some pictures of, Miss Franklin and some of the things that came from that. This is her photograph. You'll see more about that in the video later on, and kind of how they look down on it diameter-wise. So in 1953, Watson and Crick published their DNA model in a journal called Nature. Now, the internet wasn't around, right? There was no email. It's not like they posted something on their blog or vlog or put it out there and said, DNA structure like this. The journal Nature was as close to that for scientists as you could get to. Okay, any amazing, revolutionary scientific finding at the time was put in Nature. And scientists around the world in lots of different languages read their version of Nature. Okay, so this was a huge deal, and why we give credit to Watson and Crick, because they were the ones that put the DNA model out there, even though lots of scientists contributed to it. In that article, their model DNA was what we call a double helix. This is a ladder with two sides, just like a normal ladder, that's twisted. Okay, that helix is a twisted structure. They also said that strands, this is building off what Chargaff said, his rules, strands on either side of this ladder, kind of like the side parts of the ladder, and then we got rungs of a ladder, but these side parts were complementary, meaning they fit together with each other, and adenine on one side of the ladder would always pair up with thymine on the other side of the ladder, or cytosine on one side would always pair up with guanine, apple in the tree, car in the garage. Okay, this was a very detailed, at the time, look at what DNA appeared to be. We've built on to it since then and learned a little bit more, but it still holds true. That's what we understand, and that's what people use right now to sometimes change and look at DNA. And we'll see later on in another unit on biotechnology how we can modify this DNA using what it looks like according to originally began with Watson and Crick and some other scientists. The nucleotides, as we've already said, always pair up the same way. Apple to tree, car in the garage. Okay, apple to A to T and C to G. Always. I mean, you can kind of see this in this model here. So if we got the two rung or the two sides of the ladder, this twisting part of it here, these would be like the rungs of the ladder, okay, the steps of the ladder. So if there's a cytosine connected to this one, it's always going to pair up with the guanine. This is very, very important as we see later on in this unit. The backbone here is formed by covalent bonds. They connect the sugars of one nucleotide to phosphates of another. Maybe you remember, hopefully you remember back in Unit 1, covalent bonds are very strong, okay? These covalent bonds stick together through a lot of things because they share molecules, okay? Co meaning together. They link together at their molecules. The two sides, though, are held together right here in the middle by weak hydrogen bonds, okay? These hydrogen bonds are easily broken. And as we'll see, DNA many times happening right now inside your body, inside the cells, inside the nucleus of those cells, and the chromosomes that are inside those nucleus, that DNA that makes up those chromosomes is tearing apart, okay? It's ripping apart because this is how messages are carried according to the order of these nucleotides. So these hydrogen bonds need to break. They need to break and they need to get back together very easily. That's what makes hydrogen bonds so great about it. They're easily torn apart and they easily go back together. Remember the base pairing rules. I can't say it enough. Apple in the tree, car in the garage. So let's look at just some questions. Hopefully these are some you should be able to answer. Of course, there's going to be a quiz after this. What is a nucleotide? A nucleotide is the base of DNA. Okay, it's a monomer that makes up the polymer of DNA. It is a puzzle piece in the entire puzzle of DNA, or the three parts that make up one. The nitrogen bases, the phosphate groups, and the deoxyribose sugar. What are the four nitrogen bases? Look, I wouldn't say you need to know how to spell adenine or thymine, cytosine, or guanine, but be able to pick them out of a lineup, okay? Be able to recognize when you see them. Especially be able to recognize A, T, C, and G. Adenine, thymine, cytosine, and guanine. And what does Chargaff's rule state? Number 3, A always equals T. They always link together. There's always the same amount of them inside something's DNA. And C and G, the exact same. Overall shape, it's a double helix. Okay, it's a double helix. Double because there's two sides. Helix because it wraps around itself. When Watson and Crick first published their paper in 1953, they say the two strands were complementary. What does that mean? It means they always connect together in the same way. And that's according to Chargaff's rules. A to T, C to G. They're always going to link up that way. They can only link up that way. You'll never see an adenine and a cytosine linking together, okay? It's always going to be apple in the tree, car in the garage. All right, guys, use these notes to fill in your student notes and to also take some quizzes and continue on down the page.